IB Biotranscription Translation Part 4 will pick up right where we left off in Part 3, and a focus on translation. The essential idea is information transferred from DNA to mRNA is translated into an amino acid sequence. Here's a nice image that displays the 3 sites on the ribosome. We can see an arriving tRNA molecule with a glycine amino acid attached. We can see a tRNA in the P site with the polypeptide chain attached. We can see a tRNA exiting the E site. A peptide bond will be formed between the amino acid glycine on the incoming tRNA and the amino acid phenylalanine on the polypeptide chain. As the ribosome moves along the mRNA in a 5-to-3 direction, the tRNA in the P site will then move to the E site and then out of the ribosome, while the tRNA in the A site will move to the P site. Lastly, notice the triplets of codons along the mRNA are matched by complementary triplets on the tRNA known as the anticodon. We'll talk more about this later in the movie. The 2 IB syllabus statements here push us more deeply into translation. Explain the process of translation, including all components through a repeating cycle to synthesize a polypeptide. Mention initiation and termination of translation, 3 binding sites on the ribosome, and tRNA activating enzymes. State that translation occurs in a 5-to-3 direction along the mRNA. Before examining the details of the 3 tRNA binding sites on the ribosome, remember that numerous ribosomes can simultaneously translate the mRNA, and this is known as a polysome, and that translation in prokaryotic cells can begin immediately after transcription, simply because there is no nuclear membrane. In this micrograph, we can see a polysome, numerous ribosomes simultaneously translating the mRNA in the synthesis of polypeptides. In this image, we can once again see the A site, the P site, and the E site. The arriving tRNA comes into the A site, carrying an amino acid. The polypeptide chain is attached to the tRNA in the P site, and tRNA molecules exit the ribosome from the E site. The enzyme Peptidyl Transferase forms the peptide bond between amino acids, and the ribosome moves along the mRNA in a 5-to-3 direction. Remember that you need to be able to outline the structure of ribosomes, including the protein and RNA composition, the large and small subunits, the 3 tRNA binding sites, and the mRNA binding site. The ribosome is composed of 2 subunits, the large 50S subunit and the smaller 30S subunit, along with 3 binding sites A, P, and E for tRNA molecules, and a binding site for the mRNA. The enzyme Peptidyl Transferase is also part of the ribosome. The ribosome, in essence, is a large enzyme that synthesizes polypeptides. I've come back to this slide to look more closely at the relationship of the mRNA codons with the tRNA molecules that deliver the amino acids in the correct sequence. Notice the tRNA molecules have a triplet known as the anticodon that is complementary to the mRNA codons. This is the exact mechanism by which the mRNA code is translated into a correct amino acid sequence. The tRNA translates the mRNA code using the complementary nature of the codon-anticodon relationship to ensure that the correct amino acid is placed into the growing polypeptide chain. Here is an image that shows the large ribosomal subunit with its 3 binding sites. The small subunit is not shown so that more clarity is provided with respect to the tRNA molecules bound to the ribosome. You can see 2 tRNA molecules bound to the ribosome, one in the P site and one in the A site, respectively. Notice the complementary relationship of the triplet codons of the mRNA and the triplet anticodons of the tRNA molecules. The codon-anticodon relationship ensures the proper sequence of amino acids in the growing polypeptide. The ribosome moves along the mRNA in a 5-3 direction until it reaches a stop codon. This brings translation to an end. Remember the codon wheel that displays all of the possible triplets of mRNA bases. For any triplet codon, there is a tRNA molecule with the anticodon that would transport the proper amino acid to the A site of the ribosome. Also notice that there are 3 stop codons, 1, 2, and 3, that would result in translation ending as the ribosome reached the stop codon along the mRNA. In this image, we can see the mRNA with the start codon and the stop codon being read by the ribosome in a 5-3 direction. Be careful as you study this image. There are only 2 tRNA binding sites, A and P, shown here. You need to remember there is an E site. You can see the codon-anticodon relationship between the mRNA and the arriving tRNA molecules carrying the amino acids to the ribosome for the formation of a peptide bond between amino acids. As peptidyl transferase forms the peptide bond, the bond between the tRNA and this amino acid would break as the ribosome moves right, and the tRNA in the A site here is moved into the P site. This tRNA is moved to the E site, which is not shown, and then departs the ribosome. In this image, we can see a schematic diagram of the tRNA molecule, a sequence of RNA nucleotides, approximately 90 nucleotides in length. At one end, the 3' end, an amino acid will be attached by an enzyme in a process known as tRNA charging, and that involves ATP. On this end of the tRNA molecule, we can see a triplet of bases known as the anticodon. The anticodon is complementary to the codon on the mRNA, ensuring the proper positioning of the amino acid in the growing polypeptide. Here is another schematic diagram of tRNA. Hydrogen bonding holds the 3-dimensional shape of the molecule. You can see where the amino acid is attached at the 3' end, and this image nicely shows the relationship between the codon and the anticodon. The codon-anticodon relationship ensures the proper amino acid comes into place in the synthesis of the polypeptide. Here is another image of tRNA, and on the right side, a highly simplified drawing. Amino acid attachment occurs here in an enzyme-driven process known as tRNA charging, and it involves ATP. So here are the relevant IB syllabus statements. Explain that each tRNA molecule is recognized by a tRNA activating enzyme that binds a specific amino acid to the tRNA using ATP for energy. The tRNA activating enzymes illustrate enzyme substrate specificity and the role of phosphorylation. Stay tuned. In tRNA charging, an enzyme that binds ATP has specificity for one amino acid and the appropriate tRNA molecule, which will arrive here. The ATP is hydrolyzed to phosphorylate the amino acid. The amino acid will ultimately be bound to the tRNA molecule with a high-energy bond. The energy in that bond will ultimately be used to form the peptide bond as the synthesis of the polypeptide occurs. tRNA activating enzymes illustrate enzyme substrate specificity and the role of phosphorylation as ATP is hydrolyzed. The amino acid is temporarily phosphorylated with a high-energy bond that ultimately is used to form the bond between the amino acid and the tRNA molecule. The tRNA, with its bound amino acid, will then move to the ribosome, delivering this amino acid to the growing polypeptide chain. Can you identify the structures by letter in this diagram? A is the amino acid, and remember, the bond here is a high-energy bond formed at the expense of ATP by an enzyme with high specificity for the amino acid and the specific tRNA molecule with this anticodon. B is the tRNA, C is the anticodon, D is a codon, E is the mRNA, F is the ribosome, and G is the polypeptide chain. And what are the ends of the mRNA? This is the 5' end, and this is the 3' end, and the ribosome is translating the mRNA in this direction from 5 to 3. It's important for you to know that translation consists of an initiation, elongation, and termination phase, and that the synthesis of the polypeptide involves a repeated cycle of events. Initiation of translation begins with the small subunit of the ribosome, the mRNA, and the first tRNA carrying methionine as the first amino acid. While it can't be seen in this image, this tRNA will be located in the P site as the large ribosomal subunit arrives to join the small subunit. This image displays the initiation of translation with the small ribosomal subunit and the tRNA carrying methionine. But what I like about this image is that you can see the first 7 codons along the mRNA, and you can see the 5 to 3 orientation of the mRNA. The ribosome will move along the mRNA left to right, 5 to 3. In this image, the initiation of translation comes to an end with the arrival of the large subunit of the ribosome. Notice the tRNA carrying methionine sits in the P site. Elongation involves the arrival of the next tRNA to the A site. The next step in elongation is the formation of the peptide bond between the adjacent amino acids in the P and the A sites. The enzyme Peptidyl Transferase is the enzyme that forms the peptide bond. As the peptide bond is formed, the ribosome shifts right, and the tRNA in the A site moves to the P site with the attached polypeptide chain. So as I mentioned in the last slide, the ribosome shifts right. This is known as translocation. This leaves the A site open for the next arriving tRNA molecule, as the tRNA molecule that had been in the A site is moved to the P site, and it is attached to the polypeptide chain. The tRNA that had been in the P site moves to the E site and then out of the ribosome. The sequence of steps stated here happens repeatedly. As the ribosome translocates along the mRNA in a 5 to 3 direction, it will eventually reach a stop codon. There are no tRNA molecules that match the stop codon. Translation ends with release of the mRNA from the ribosome and a release of the newly formed polypeptide. Remember that translation in the prokaryotic cell can occur immediately after transcription due to the absence of the nuclear membrane. And that brings us to the end of Transcription Translation Part 4. In Part 5, we will review translation and look briefly at mutations.